Hello, everyone, and welcome to today's session. And today we'll be discussing the theory for the chapter Biological Classification. So, this is the part one of the lecture. Before starting this lecture, I just want to give you one information that the images, text, and other materials used in the presentations are for education purposes only. We respect the intellectual property rights of the original creators and owners. If you are the owner and have any concerns or issues, please contact us at sathee@itk.ac.in and we shall remove the content immediately. Please note that parts of the content may have generated using generative AI, which is experimental. So let's begin with the learning objectives now. So first, we will understand the Whitaker's 5 kingdom classification. And we'll also understand the criteria for the Whitaker's 5 kingdom classification. Okay, which means that how Whitaker classified the different living organisms into various kingdoms. What was the basis which Whitaker used for classifying different organisms into these different kingdoms. Then we'll talk about the characteristic feature of the 5 kingdoms, Monera, Protista, fungi. Okay, so basically we'll talk about Monera, Protista and fungi. Okay. Yes, and we'll also talk about little bit in about the kingdom Plantae and kingdom Animalia that we'll study in the next session. Okay, today we'll focus on the kingdom Monera. Clear? Then we'll also talk about the different types of bacteria. Okay, how we can group bacteria based on their shape. We'll talk about this. We'll also talk about the archaebacteria and the eubacteria. Okay, so we'll talk about the introduction. I will introduce you to this chapter, biological classification. We'll talk about kingdom Monera, and within kingdom Monera, we'll talk about archaebacteria and eubacteria. Okay, so here you can see the earlier classification system. Clear, the earlier classification system was just based on the use of the, you can say, animals. So a lot of animals, they were beneficial to the human beings. Clear? So the earlier classification system, they were based on the use of the animals. Then the classification system changed. Then one of the first classification system was given by Aristotle. So Aristotle classification had some scientific basis, and he divided the living organism into 2 groups. One was the organism containing red blood cells, or you can say red blood. Okay, and other group he made, okay, in which the organism didn't consist of the red blood. Okay, so he divided animals into 2 groups. Okay, one of the group consisted of the organism that contains red blood, and the other group consisted of the organism that didn't have the red blood. Similarly, he divided plants also. Okay, clear? So this had a little bit of scientific basis. Clear? Now there was a modification in the classification system. So earlier the classification system was based on certain morphological characteristic, means external characteristic. Later on, these other features was also included, like cell wall structure, mode of nutrition, mode of reproduction, evolutionary relationship. So the classification system were changing because we were including new features now. So criteria of the classification was changing. Okay, so you have to remember the classification system given by Aristotle. Clear? And the plants was divided into herbs, shrubs and trees. Now, these changes will happen in future also. This will take place in future also. Okay, because we are including new features now. We are including features like I've already told you, the cell wall, the nature of the, you can say, reproduction. Okay, evolutionary relationship we are including. Clear? We are including a lot of, you can say, molecular data now. Clear? Genetic studies is being done. Now the technology has been improved. We have better microscopes. We have better technology. So we can better understand the organisms now. Okay, so that's why the classification is changing, and it will change in future also. Clear? Now, the evolutionary, or you can say the classification system, or the modern classification system, these are based on morphological, physiological, reproductive similarities. Okay, and these also include evolutionary relationship. So these are the basis of the, you can say, of the modern classification system. They not only include morphological, physiological, and reproductive similarities, they also include phylogenetic. Phylogenetic means evolutionary relationship. Now in this chapter, we'll study characteristic of kingdom Monera, Protista, and fungi. Okay, so we'll focus on these three kingdoms. There are other kingdoms also, like kingdom Plantae, kingdom Animalia. Clear? But in this lecture, we'll focus on kingdom Monera. Okay, kingdom Plantae and kingdom Animalia. We'll also talk about these, but in the other chapters, plant kingdom and the animal kingdom. Okay, so you should know about the characteristic features of kingdom Monera, Protista, fungi, Plantae, and Animalia. So if we talk about kingdom Monera, what are the features of kingdom Monera? So in kingdom Monera, all the prokaryotes are included, and these are unicellular, means single celled. And if we talk about the cell wall of kingdom Monera, so cell wall is usually made up of polysaccharides and amino acids. Clear? Then they are unicellular, means they are single celled. Clear? If we talk about the reproduction, they reproduce by asexual methods, which is binary fission. Okay. If we talk about their mode of nutrition, so these can be phototrophic, these can be chemosynthetic bacteria or photosynthetic bacteria, or they can be heterotrophs also. When we talk about phototrophic bacteria, this means they can photosynthesize and they can prepare their own food. So what happens in photosynthesis? Carbon dioxide combines with water to form glucose in the presence of chlorophyll and sunlight. So the members of kingdom Monera, which includes bacteria which are prokaryotes, these can be photosynthetic and these can be chemosynthetic also. Clear? Chemosynthetic means they oxidize the inorganic substances to produce energy. Example of chemosynthetic bacteria can be like nitrosomonas or sulfur oxidizing bacteria. Then the members of kingdom Monera can be heterotrophs also, means they obtain nutrients from other organisms. And again, heterotrophs can be of two types. One is saprophytes, another one is parasitic. So saprophytic organisms, they feed on or they obtain nutrients from dead and decaying organic matter. If we talk about the parasitic organisms, these parasitic organisms, they are parasites on the host, so they obtain nutrients from the host. Okay, then coming to kingdom Protista. Kingdom Protista, you have to remember, includes all the unicellular eukaryotes. Prokaryotes are only included in kingdom Monera. From Protista onwards, you have all the eukaryotes. Fungi also include eukaryotes. Plant also include eukaryotes. Animalia, these are also eukaryotes. Okay, so you have to remember, Protista includes unicellular eukaryotes. So what is the difference between prokaryote and a eukaryote? Prokaryotes, they lack the true nucleus. They lack the nuclear membrane. They lack the cell-bound, membrane-bound cellular organelles. And if you talk about the eukaryotic cells, they have true nucleus. Okay, they have membrane-bound cell organelles. Here, so Protista include single-celled or unicellular eukaryotes. Then you can see protist. They can be also heterotrophs or autotrophs. Clear? Then they are also single-celled. Like members of Kingdom Monera, Protista are also single-celled, means they are unicellular. Clear? Then if we talk about the fungi, if we talk about this cell wall of fungi, it is made up of chitin. Then again, fungi, they are also eukaryotes. Okay. Clear? What is the mode of nutrition in fungi? So they are saprophytes, means they are heterotrophs. They feed on dead and decaying organic matter. If we talk about Kingdom Plantae, again they are eukaryotes. Okay, cell wall is made up of cellulose. And if you talk about Kingdom Animalia, again they are eukaryotes. And there is no cell wall in animals. Okay, animals, they are heterotrophs. Plants, they are always phototrophs. They can photosynthesize. Plants have chlorophyll in them. Okay, chlorophyll is the primary pigment. Chlorophyll A basically is the primary pigment which helps in the photosynthesis. Okay, chlorophyll A molecule absorbs the sunlight and converts light energy into chemical energy. Similarly, cyanobacteria, which is blue-green algae. Okay, this also consists of chlorophyll A, which is similar to the chlorophyll A of the green plants. So that's why members of Kingdom Monera, they were photoautotrophs also. They contain chlorophyll A also, which is quite similar to the chlorophyll A of the green plants. Okay, so you should remember the characteristic features of Kingdom Monera. Okay, Protista, fungi, plantae and animalia. Clear? So we'll basically talk about the 5 kingdom classification in this chapter. So there are other classification systems also. Like there was 2 kingdom classification given by Carolus Linnaeus. Okay, what were the 2 kingdoms given by Carolus Linnaeus? They were Kingdom Plantae and Kingdom Animalia. But there were limitations to the 2 kingdom classification. Okay, so what were the limitations of the 2 kingdom classification? Let's discuss this briefly. So firstly, first limitation was that this classification system was not able to distinguish between prokaryotes and eukaryotes. Clear? Then this system was not able to distinguish between unicellular and multicellular. Okay. Clear? Multicellular organisms also included here and unicellular organisms were also included here. Then this system was not able to distinguish between the heterotrophic organisms and the phototrophic organisms. All the organisms, they were included in one group only. Like Kingdom Plantae, it included fungi also. Fungi, you know, it is a different group. It included bacteria also. It included algae also. Plants also. And what was the basis of putting all these organisms into Kingdom Plantae? Basis was that all were having the cell wall. Okay. But you know, all these organisms, fungi, bacteria, they are different in other characters also. Okay. Clear? Like fungi, they have a cell wall made up of chitin. Plants, they have a cell wall made up of cellulose. Bacteria, they have a cell wall made up of peptidoglycan. Clear? So there was limitation to 2 kingdom classification. Then let's talk about the 3 domain system. So these 3 domain, what happened in the 3 domain classification system? The Kingdom Monera were further divided into 2 domains. So Kingdom Monera consists of all the prokaryotes. Okay. So what were the 3 domains? Archaea, bacteria and eukarya. These were the 3 domains. Archaea, this included all the single-celled prokaryotic organism. Then bacteria also, Okay, bacteria included the true bacteria. The archaea included the bacteria which lived in the extreme environment. Okay, that's why the name archaea. Archaea included all the single-celled prokaryote, and the bacteria included in archaea, these used to live. These lives in the extreme environments, so they are known as extremophils. Okay, then after archaea, there was another domain, bacteria. Bacteria included the true bacteria. So these were also single-celled. Okay, so domain bacteria included the true bacteria. Then the third domain, eukarya, it included all the eukaryotic organisms. Okay, so protista, fungi, plantae and animalia, these were all included in the domain eukarya. Now you can see this can be also called as 6 kingdom classification system, as Monera is divided into 2 groups. Okay, so this can be also called as 6 kingdom classification. Okay, so again there were 3 domains, archaea, bacteria and eukarya. Archaea and bacteria, this included the prokaryotes, single cell prokaryotes. Okay, archaea included the extremophils, and bacteria included the true bacteria, and eukarya included all the eukaryotic organisms. Clear? Now, in this session, we'll discuss kingdom Monera in detail. Okay, so let's start with this. Bacteria, they are the sole members of the kingdom Monera. Bacteria, you know, they are the prokaryotes. Mean prokaryotes are included in the kingdom Monera. Clear? What are the features of prokaryotes? Prokaryote, they lack the true nucleus. Means they don't have a nuclear membrane. Clear? So they lack the true nucleus. Instead of the nucleus, they have nucleoid. So what is nucleoid? Nucleoid is the irregularly shaped region that consists of the genetic material. In case of bacteria, what is genetic material in bacteria? DNA. So DNA is not enclosed, it is not present within the nucleus. So there is no true nucleus. Instead of nucleus, there is a nucleoid. Nucleoid is a region where the genetic material is present. What are the other features of prokaryotes? It consists of, okay, it doesn't consist of cell-bound or membrane-bound cell organelles. So bacteria, which is a prokaryote, they are the sole members of the kingdom Monera. Okay, they are the most abundant microorganisms. So bacteria, they are most abundant microorganisms. They are present everywhere. Clear? They are present in hot springs also. They are present in the very cold temperature also. They are present in deep oceans also. Clear? So bacteria occur everywhere. They are cosmopolitan. And hundreds of bacteria are present in handful of the soil. So if you pick up the soil, there will be millions of bacteria in it. There are bacteria on your hands. There are bacteria on your skin. Bacteria are everywhere. They are even present in the air. They can be present in the form of spores. Now they live in extreme habitat also. Some of the bacteria, they can survive in extreme habitat also, such as hot springs, desert, snow and deep oceans, and very few other life forms can survive. So you can see these are quite extreme environments, extreme habitat. In hot spring, the temperature is very high. Again, desert, the temperature is high. There is low moisture in the air. Okay. Then snow. Snow means temperature is very less. Then deep oceans. This is also extreme environment here. In deep oceans, there is no sunlight. Now, many of them live in or on other organisms and parasites. Some of the bacteria can also be parasites. So parasites, they obtain nutrients from their host. So parasite, they can live in the host or on the host. Clear? And they obtain nutrients from the host. So in parasitic interaction, the parasite is benefited, but the host is, you can say, is not benefited, or host is harmed, because the parasite is obtaining nutrients from the host. Now, bacteria are grouped into 4 categories based on their shape. So let's talk about the 4 categories of bacteria based on their shape. First is the spherical. The bacteria can be spherical in shape, for example, coccus. It can be rod shaped, such as bacillus. Then they can be comma shaped, such as Vibrio, and they can be spiral shaped, such as Spirillum. So these are the different shape of the bacteria. Here, the bacteria can be grouped, or bacteria can be divided into various groups based on their shape. So you should remember this. Now, though the bacterial structure is very simple, they are very complex in their behavior. Okay, so bacteria, they are lower organisms. They have a very simple structure. In bacteria, what is happening? All the activities is done by a single cell. So they are unicellular, they are single celled. Okay, a single cell is performing all the functions: respiration, metabolism, excretion, everything is being performed by a single cell. Clear? So they have very simple structure, but they have a very complex behavior. Okay, so you can see these are bacteria of different shape. So bacteria can be spherical. Okay, bacteria can be rod shaped. Example is bacillus. Then they can be spiral shaped, and they can have flagella also. Flagella helps in the motility of the bacteria, and they can be comma shaped, such as Vibrio. Example is Vibrio cholerae, which cause cholera. Now, compared to many other organisms, bacteria as a group show most extensive metabolic diversity. So, a lot of biochemical reactions occur in bacteria. So, bacteria, they can survive in in extreme habitat also, and they are present everywhere. So they are very diverse metabolic. They have extensive metabolic diversity, which helps them to survive in different environments. Now, some of the bacteria are autotrophic, and they synthesize their own food from inorganic substrates. So autotrophic means, auto means self. So they are autotroph, they can synthesize their own food from inorganic substances. Like in photosynthesis, what happens? Inorganic substances like carbon dioxide and water, they combine together in the presence of chlorophyll and sunlight to produce carbohydrates or glucose. Clear? So bacteria, they are autotrophs. Now, they may be photosynthetic autotrophs or chemosynthetic autotrophs. Photosynthetic autotrophs means they can photosynthesize. They are photosynthetic bacteria. Like cyanobacteria, it can photosynthesize. It is known as blue-green algae. It consists of chlorophyll A, which is very similar to the chlorophyll A of the plants. Then there is a chemosynthetic autotrophs. So chemosynthetic autotrophs, they can oxidize the inorganic substances to produce energy. Examples are nitrosomonas or sulfur oxidizing bacteria. Now, majority of the bacteria are heterotrophs. What are heterotrophs? Which obtain nutrients from other organisms, or which are dependent on other organisms for nutrition. Okay, so they depend on other organisms or on dead organic matter for food. So when we talk about heterotrophs, within heterotrophs, there are two things, okay, or two categories you can say. First one is the parasitic. Second one is the saprophytic. So parasitic is when the bacteria is a parasite on the host. It obtains nutrients from the host. Next one is saprophytic. In saprophyte, what happens? The saprophyte, they feed on the dead and decaying organic matter. They obtain nutrients from the dead and decaying organic matter. So they can secrete enzymes, digestive enzymes, which can degrade the organic matter, and then they absorb the nutrients from this degraded organic matter or decomposed organic matter. Okay, now Kingdom Monera again have these two subgroups. First one is the archaebacteria. So archaebacteria, they are the primitive bacteria. They live in extreme environments. That's why they are known as extremophiles. Okay, so they can live in hot springs, deep ocean vents. Okay, deep ocean vents. They can live in high saline conditions. Now, these bacteria are special since they live in some of the most harsh habitats, such as extreme salty areas. Okay, they are known as halophiles, which live in extreme saline conditions. Okay, they can live in hot springs where the temperature is very high. They are known as thermoacidophiles. They can live in marshy areas. Clear? And they are the bacteria which live in marshy area. They are known as methanogens. Clear? Now, archaebacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for their survival in the extreme conditions. So, what were the extreme conditions? High saline condition, hot springs, and then marshy areas. Clear? The bacteria which live in hot spring, they are known as thermoacidophiles. And methanogens, they have the capacity to produce methane gas. So, how archaebacteria can survive in this extreme environment? So, answer is in their cell wall structure. So usually the bacteria have peptidoglycan in their cell wall. But archaebacteria, the cell wall of the archaebacteria is quite different. The cell wall, what is the component of the cell wall, or what is the composition of the cell wall of the archaebacteria? So the cell wall consists of pseudomurine instead of the peptidoglycan. It can also contain certain glycoproteins, okay, certain complex polysaccharides. Okay, so there is no peptidoglycan. Instead of peptidoglycan, you have pseudomurine or glycoproteins or some complex polysaccharide. So cell wall is different, and due to this cell wall, they are able to survive in the extreme conditions. Now let's talk about the methanogens. So what are methanogens? Okay, as the name suggests, methanogens, they can produce methane gas. Okay, and where are methanogens present? They are present in the gut of several ruminant animals such as cow and buffaloes, and they are responsible for the production of methane gas, which is a major component of the biogas. Biogas is the biofuel. Okay, so this methane can be produced from the dung of these animals using methanogens. Clear? So methanogens come under archaebacteria. Now let's talk about eubacteria. So eubacteria, they are the true bacteria. Clear? So there are thousands of different eubacteria or true bacteria. Archaebacteria, they are the primitive bacteria. And they are characterized by the presence of rigid cell wall, and if motile, they consist of the flagellum also. The cell wall of eubacteria is different from that of archaebacteria. So here the cell wall consists of peptidoglycan. And they also consist of a flagellum, which helps them to move. So they help in the motility of the eubacteria. Clear? The peptidoglycan, which is a component of the cell wall of the eubacteria, this is a complex carbohydrate. Clear? Let's talk about cyanobacteria. They are also known as the blue-green bacteria. They have chlorophyll A similar to green plants and are photosynthetic autotrophs. So again, question can come from this line. Okay. What is the mode of nutrition in cyanobacteria, also known as blue-green algae? So cyanobacteria, they are photosynthetic autotrophs because they have chlorophyll A. So chlorophyll A, if we talk about plants, it is a primary pigment in plants also, which helps to capture the light energy. Clear? So this chlorophyll A, which is quite similar to the green plants, is present in cyanobacteria also. So it can photosynthesize. It can convert light energy into chemical energy, or it can convert, you can say, light energy simply into food. That's why it is known as photosynthetic autotrophs. It can prepare its own food using light energy or using light. Cyanobacteria, they are unicellular, or they can be colonial or filamentous. They can live in fresh water or marine water, or they can be terrestrial also. Okay, so this is an image showing you filamentous blue-green algae. So you can see this is nostoc. Nostoc is a filamentous blue-green algae, means it is a cyanobacteria. What is special about nostoc? First of all, it consists of a specialized cells known as heterocyst. So within this heterocyst, nitrogen fixation can take place. Thus, atmospheric nitrogen fixation can occur in the heterocyst. Again, question can come from here. What is the function of heterocyst? It also consists of a mucilaginous sheath around it. Here, so it is a blue-green algae, means it can photosynthesize. It consists of chlorophyll A pigment. The colonies are generally surrounded by gelatinous sheath, as shown in the figure. They often form blooms in the polluted water bodies. Okay, so what does this mean? They form algal blooms in polluted water bodies. So what happens? Due to excessive nutrients, especially nitrogen and phosphorus, there is extensive growth of the algae. This is known as algal bloom. Now, as the algae dies, okay, a lot of organic matter will be accumulated, and this algae will now decompose, and for decomposition oxygen is required. Okay, so now decomposition of the algae is utilizing the oxygen, so this increases the BOD, means biological oxygen demand, for the other aquatic animals. So it can be dangerous to other aquatic animals because there will be lack of oxygen for them, because oxygen is being used for decomposing the algae. Okay. Now, some of these organisms can fix atmospheric nitrogen in specialized cells called heterocyst. We've already talked about heterocyst. Okay, what are the examples? Nostoc and Anamina. So they have heterocyst in which they can fix the atmospheric nitrogen. And let's talk about the chemosynthetic autotrophs. So autotrophs can be of two types, photosynthetic autotrophs or chemosynthetic autotrophs. What are chemosynthetic autotrophs? So these bacteria can oxidize various inorganic substances such as nitrates, nitrites and ammonia, and use the release energy for their ATP production. Okay, example is sulfur oxidizing bacteria. Okay, or they can also utilize nitrates, nitrites and ammonia. Okay, they can oxidize these inorganic substances. Okay, and they can use the release energy for ATP production. Now, they play a great role in recycling nutrients like nitrogen, phosphorus, iron and sulfur. So again, you should remember this chemosynthetic autotrophic bacteria, they help in the nutrient cycling. Okay, as they use inorganic substances for energy production. So in this process, okay, what they do? They help in the nutrient recycling. Nutrient recycling is very important. Okay, you can say all the nutrients they again return to the soil and they are absorbed by the plants. And then again the plant will die. Okay, the organic matter will be decomposed. These bacteria will act on the you can say decaying organic matter. This organic matter will be converted into inorganic substances. Okay, and then mineralization will take place. Okay, basically nutrient recycling is occurring here. Now, earlier I told you that the members of Kingdom Monera, they can be autotrophic, they can be autotrophs, or they can be heterotrophs. We have already talked about the autotrophs. Within autotrophs, we talk about photosynthetic autotrophs and chemosynthetic autotrophs. Now we'll talk about the heterotrophic bacteria. So heterotrophs means organism which derive nutrients from the other organism or which are dependent on other organisms for nutrition. And within heterotrophs, there are two categories. One is parasitic, another one is saprophytic. So heterotrophic bacteria are most abundant in nature, and the majority are important decomposers. So they are known as decomposers. They decompose the organic matter. Now, most of them have a significant impact on human affairs. Let's understand this. Some are helpful in making curd from milk. So, lactobacillus, which is lactic acid bacteria. This can convert milk into curd. Then they produce a lot of antibiotics also. Clear? Like streptomycin is an antibiotic. Then they are also involved in fixing nitrogen in leguminous plants. So they can form nodules there. Clear? So these are some of the benefits of these bacteria. They can be used for human welfare as well because they are producing antibiotics. Some are pathogens also. Clear? So pathogens, what are pathogens? Organisms which can cause disease. So some are pathogens and they cause damage to human beings because they cause a lot of diseases. They can damage crops also. Again, they can cause disease in crops. They can cause damage to animals as well as the pets also. Like cholera, which is caused by Vibrio cholerae. This is a disease caused in humans. Then typhoid caused by Salmonella typhi. Okay, then tetanus, citrus canker. They're all well-known diseases caused by different bacteria. Now, bacteria reproduce mainly by fission. So this is a kind of asexual reproduction. In fission, what happens? If we talk about binary fission, one cell divides into two cells. So this is a parent cell. This parent cell will divide into two daughter cells. And the two daughter cells which are produced, they are genetically identical to themselves also, and they will be genetically identical to the parent, means they are clones. Now, sometimes under unfavorable conditions, bacteria may produce spores. So this is a kind of survival strategy. And then when favorable conditions, they reappear, means when there is optimum temperature, optimum, you can say, conditions, optimum pH, okay, optimum moisture conditions, then the spores can again germinate. Clear? And then again it can form a vegetative cell which is capable of growth and reproduction. Now they can also reproduce by sort of sexual reproduction. Important here is to understand sort of sexual reproduction. By adopting a primitive type of DNA transfer from one bacterium to another. So there is they are saying this is a sort of sexual reproduction. This is not a true sexual reproduction. This is a sort of sexual reproduction where the bacteria can transfer its DNA to other bacteria. So this is known as horizontal gene transfer. Transfer of the DNA within one generation from one bacteria to another bacteria. Because you must have heard about conjugation. Conjugation, what happens? One bacteria, it forms a sex pilus, and through this sex pilus, the two bacteria gets connected, and then one of the bacteria transfers DNA into the other bacteria. Okay, so this is a dividing bacterium. One cell is dividing into two. This is a cell wall of the bacteria. This is a cell membrane. And this is the DNA, which is the genetic material of the bacteria. The one cell is divided into two daughter cells. Okay, let's talk about the mycoplasma. Mycoplasma, they lack the cell wall. So because they lack the cell wall, so that's why they are the smallest known living organisms and they can survive without oxygen. Mycoplasma, they are pathogenic in animals and plants. They can cause a lot of diseases in plants and animals. Let's talk about, okay, let's now summarize the entire theory that we have discussed. We talked about the five kingdom system of classification given by Whitaker in 1969. So he divided the life forms into Monera, which includes bacteria, then Protista, which includes single cell eukaryotes. Then fungi also are eukaryotes, plant are also eukaryote, animals are also eukaryotes. What was the criteria used by Whitaker? So he grouped different organisms into various kingdoms based on cell type, body organization, mode of nutrition. Okay, clear? Then mode of reproduction. Now we have also talked about the two kingdom system classification given by Linnaeus in 735. So he divided the living organisms into two kingdom Plantae and Animalia, but there were a lot of limitations of two kingdom classification. Then there was a three domain system given by Wies in 1990. Okay, what were the three domains? Bacteria, archaea, and eukarya. Now we talked about the kingdom Monera. So this consists of the prokaryotic organisms. Okay, it consists of the true bacteria and the archaebacteria. They are single cell, means they are unicellular. They lack the nuclear membrane. They reproduce by binary fission and they are found everywhere. Okay, so this was the end of the theory. Let's start with the DPP of this chapter. Okay, so this is the DPP that we are going to discuss now. Let's start with the first question. Which of the following bacteria is responsible for producing methane, which is a major component of the biogas from the dung of the ruminant animals? Okay, so which bacteria have the capability to produce methane from the dung of ruminant animals? What are the options? Halophiles, thermoacidophiles, methanogens, and cyanobacteria. What will be the correct answer? Okay, here the correct answer will be methanogens. Methanogens, they have the capability to produce methane, which is methane is a major component of the biogas. So they are usually present in the gut of the ruminant animals such as cow and buffaloes. Now let's discuss this question. Which characteristic differentiate archaebacteria from other bacteria? So you know archaebacteria, they are known as extremophiles. They can survive in extreme environments. So how they can survive in extreme environments? Okay, or what differentiates archaebacteria from the other bacteria? But the option presence of chlorophyll A, option B is different cell wall structure, C is ability to form heterocysts, and D is reproduction via binary fission. So what will be the correct answer? The correct answer is different cell wall structure. So archaebacteria, the cell wall of archaebacteria, they consist of pseudomurine, some complex polysaccharides or glycoproteins. Whereas the eubacteria, the cell wall of the eubacteria consists of peptidoglycan. So there is no peptidoglycan present in the cell wall of archaebacteria. The next question, cyanobacteria, also known as blue-green algae, are classified as chemosynthetic autotroph, photosynthetic heterotrophs, and photosynthetic autotrophs, and parasitic organisms. So what will be the correct answer here? So you know autotrophs, they can prepare their own food. Heterotrophs, they are dependent on other organisms. Then autotrophs can be of two types, photosynthetic or chemosynthetic. Heterotrophs can be of two types, parasitic or saprophytic. So what will be the correct answer here? Cyanobacteria, you know, they consist of the chlorophyll A, which is quite similar to the chlorophyll A of the green plant. So answer should be, they are photosynthetic autotrophs. Let's discuss this question. Which structure is absent in the mycoplasma? So mycoplasma, they are the smallest living organisms. Why they are smallest living organisms? So what are the options here? Cell membrane, flagellum, cell wall, and DNA. Here the correct answer will be cell wall. So mycoplasma, they lack cell wall. That's why they are the smallest living organisms. Next question, heterocysts in cyanobacteria are specialized for. Okay. What is the function of heterocyst? First option, photosynthesis, nitrogen fixation, spore formation, or DNA transfer. So the answer is nitrogen fixation. Okay. So we have completed the DPP also. Okay. So all our answers are correct. So this was the end of the session. I hope this session was quite knowledgeable for you. Thank you very much for attending.